Hello everyone, my name is Madhu, a PhD student in the Department of Oncology at the University of Alberta. My PhD project focuses on studying about the cancer called chronic lymphocytic leukemia, also known as CLL.
CLL is the most common type of adult leukemia. It's a cancer of white blood cells. And although treatments have improved over the years, many patients still relapse because cancer cells become resistant to therapy. One reason this happens is because cancer cells are incredibly good at adapting and surviving. They hide in the protective environments inside the body, especially in the bone marrow, where they receive support from the surrounding cells. You can think of it like cancer cells building a shelter that protects them from treatment. Our research focuses on finding a way to break that protection. Inside every cell, there is a structure called the lysosome. Lysosomes act like the recycling and waste management system of the cell. They help cells survive stress by cleaning up damaged materials and recycling nutrients. But cancer cells hijack this system to help themselves survive. Our research investigates whether we can target lysosomes to weaken leukemia cells and make them more vulnerable to treatment. In particular, we study a drug called pimazide. Interestingly, this drug was originally developed for neurological disorders, but we discovered it may also disrupt lysosomes in leukemia cells. When lysosomes stop functioning properly, the cancer cells experience stress, accumulate damage, and may eventually die.
What makes this special is that we study this in a 3D spheroid model, a miniature model that better represents what actually happens inside the human body.
Instead of growing cancer cells flat on a plastic dish, we grow them together with supportive bone marrow cells in a three-dimensional structure. This allows us to recreate the protective environment that leukemia cells normally hide in.
Using this model, we can ask important questions. Can lysosome targeted therapy break the communication between the leukemia cells and their protective microenvironment?
So far, our results suggest that disrupting lysosomes not only stresses the leukemia cells themselves, but may also interfere with the signals that help them survive. This is important because overcoming drug resistance remains one of the biggest challenges in cancer treatment.
Ultimately, our goal is to develop effective therapies that prevent the leukemia cells from escaping the treatment and returning later.